In this module, the role of microRNAs in the translational level control will be discussed, and we will study that how microRNA play their role in this control. Now we know that the proteins are not the only molecules that can act as regulator of messenger RNA. Remember, proteins, they are not the only molecules which can act as regulator for messenger RNA translation and the stability. Messenger RNA translation and stability is very important. And there are other molecules which are also play a part in it. MicroRNAs, they primarily act by binding to the sites in the 3 UTR of their target messenger RNA. That on the 3' site, the UTR which are there, that is the binding sites for messenger microRNAs. Remember, in the case of messenger RNAs. Now it is becoming increasingly clear that microRNAs are important translational level regulator of virtually every biological process. So microRNAs, they also take part in the translational level regulation of biological process. Now, what are biological processes? If we consider biological processes are the synthesis of various proteins, synthesis of various macromolecules for the cell requirement, what it is. Now, microRNAs, they likely exert their regulatory activities through several distinct mechanisms. They are different mechanisms through which the microRNAs, they take part, and that mechanism, they affect the messenger RNA translation and messenger RNA stability. That is two both important part. That is the messenger RNA translation and messenger RNA stability. Of course, if messenger RNA is stable, then its messenger RNA translation will also be active. Now, how microRNA control gene expression? What are the different mechanisms by which the microRNA, they control the gene expression? The one is the microRNA, they can express the synthesis of proteins. They can express the synthesis of protein at some point after the initiation of translation has occurred. When the initiation has occurred, what microRNA can do, it can repress the synthesis of proteins. Specific protein, it can repress that. RISC, which is abbreviation of RNA-induced silencing complex. So this is that at that point, that gene or the synthesis of that protein is suppressed. It means that gene is silenced. And RISC, it is believed to cause premature drop-off ribosome. That when through RISC, what happened? That the premature ribosomes where the synthesis of protein occurs, that is drop off. Another study suggests that due to bound RISC interferes with translation initiation. One is that at certain point of initiation it acts, but the second is that it interferes with translation initiation. That is, it stops or it hinders or it inhibits the initiation of translation of the messenger RNA. Just shown in this diagram, there are these two different processes which has been illustrated, where what happened that the translation represses RISC, that it is repressing the translation. Translation cannot take place. Whereas in the other side, what happened, RISC, the messenger RNA, it stops and it cannot create or it cannot synthesize that protein because ribosomes, they drop off and the process is ceased or process stops. Now, still other studies, they have suggested that some microRNAs, they carry out translational level regulation by inducing the degradation of the target RNA. So as we see in the previous diagram, that the microRNA, they have carried out the translational level regulation at the translational level, that is when the cell is ready, when the messenger RNA is ready to translate for the protein, at that level, the microRNA, it induces the degradation of target messenger RNA because it induces a process where messenger RNA is degraded. So if there is no messenger RNA, so it means it is controlling the synthesis of the genes at that level. So in this module, we have discussed the different level, the different processes where microRNA can play a part for the control of the synthesis of the gene at the gene level and the various processes where it is activated.